Good morning, everyone. Thank you for joining us. I'm Monica Coenraads. I have a 29-year-old daughter with Rett syndrome who was diagnosed at the age of 2. For the past 26 years or so, I've been raising funds and working with the scientific and clinical community to advance therapeutics for Rett syndrome. In 2009 or so, we had a number of families approach the Rett Syndrome Research Trust, the organization that I started, and asked whether we could also fund research for duplication syndrome if the families fundraised. And we also had the same request from Dr. Zogby and others. And we agreed because, one, we felt it was the right thing to do. But secondly, because there's a lot of overlap in terms of some of the basic science and the investigators and clinicians. And so we thought it was a good fit. So the community has raised some money, and over the years, we have funded a number of projects, including the reversal experiment that Dr. Zogby did, and the research that she did that has led to the UMASS trial. And so that's kind of the backstory of why the Rett Syndrome Research Trust is involved with duplication. I met Tim Riley a number of years ago. He was actually our chief scientific officer for a number of years. And he brought on a number of researchers at UMASS Medical School, including Anastasia Khvorova, to work on Rett syndrome and to work on duplication syndrome. Through Tim, I then met Ken Moch. And of course, you all know Dr. Pehlivan, who runs the clinic at Baylor. So I'm gonna let each of them introduce themselves. Let me just share my screen. Okay, Tim, I'm actually going to start with you. If you could just give a short little background about yourself and just how you started getting involved with duplication.
Great, thanks, Monica. About 35 years of drug development experience in early stage and mid-stage pharmaceutical companies, including lots of international experience. I was on the executive committee of a publicly traded company for about 10 years. I got involved with duplication, as Monica pointed out. I went to Anastasia and said, I think siRNAs would be perfect. They're potent. They'd be great to knock down duplication. She had an ambitious, very competent postdoc whose name was Vignesh Hariharan, and he took it on with zeal and made great progress. So that's really how I got involved with duplication. We'd also got some other programs funded for other things, such as a different type of transgenic mouse up at Kids, right, in Toronto, Monica?
With kids, yeah.
Yep. So that's kind of my background in duplication, and I've sort of kept tabs on it. And then, as Monica pointed out, I brought Ken Moch, who's a strong business colleague and partner, into the mix.
Okay, Ken, if you could introduce yourself.
Sure, and great to meet everybody. And indeed, Tim and I have known each other for many years, never actually worked together, but crossed paths on one of the companies that I ran and that he was at before I was there. Scientific background in college, went to business school, was at McKinsey in Chicago, and got pulled out to be the first business person in the first lipid nanoparticle company, cleverly called the Liposome Company, more years ago than I care to admit, and raised the money and helped take it public. Then launched the first cord blood stem cell company, now about 35 years ago. I've run companies in the biotech space in a variety of different therapeutic categories. Alzheimer's, antiviral therapeutics, which is the one that Tim and I worked on, that has a drug approved for smallpox, which I pray is never used, cardiovascular diabetes. And Tim and I were talking now, I guess about 4 or so years ago, about various things that he was involved with, and he introduced me to the folks at UMass, and that's what led to to my involvement here. So really delighted to meet everybody by Zoom, and kind of thankful to Tim for introducing me to the whole area.
Great. Dr. Pehlivan.
Hi, everyone. I am Davut Pehlivan. I am a clinical neurogeneticist, but I have bench work experience in genetic disorders, especially MECP2 Duplication Syndrome in Huda Zogby and Jim Lapsky's lab. As you know, I am doing translational research and the clinical trial for the MECP2 Duplication Syndrome. I think RNA-based treatments are a great approach for MECP2 Duplication Syndrome. We are thankful that we have ASO trial going on now, but we need to have more options, more treatment options for the MECP2 duplication world. That's why I am part of the team now at M2DS to hopefully bring this siRNA approach to the clinical trial level as well. So that's all about me.
Okay, great. I'm going to stop sharing, and Tim, who's going to do present the bulk of the presentation today, he's going to pick it up from here. By the way, we're going to do Q&A at the end, but by all means, enter your questions as they pop into your head, and we'll do our best to address them at the end of the webinar.
Monica, it's recording, right?
Yes, it's recording, and the recording will be available as well.
Can everyone see
Yep.
my screen now?
It's up, Tim.
Excellent. Great. We've called the company, and it is a formal company, M2DS Therapeutics. And you've already met all of us, so I won't belabor that anymore. Today's agenda, we're going to be introducing who actually did the research behind all this, what really the mission is. I'll describe it to you briefly what the technology is, and kind of tell you what the status of where we are as the development of the program, as well as Ken will share where we are from a business point of view for the program. We'll talk a little bit about company creation in biotech. Some of you may be familiar with that, some may not. We'll go a little bit into the market size. We realize that this is always a moving target a little bit, and as Monica's want to point out, these numbers usually go up once there is a treatment. So, but from a business point of view and in talking to investors, we have to sort of do comparables and peg it against other indications. We'll talk a little bit about the competition as well, and where we're looking for our projected milestones and any final thoughts, and the funds that we need really to move it forward, and what we're trying to do around that, and any final thoughts on the program and your questions. I don't have to tell you what MECP2 duplication is, but our goal is indeed to reduce the primary symptoms for MECP2 duplication. So UMASS Chan Medical School in Worcester has become a real powerhouse for RNA therapeutics. They have the RNA Therapeutics Institute. They have Gene Therapy Institute as well. Craig Mello got the Nobel Prize for siRNA, and then Victor Ambrose, just this last year, got the Nobel Prize, shared the Nobel Prize for microRNAs, both of which are related to each other, siRNAs and microRNAs. So it's just a wonderful institution to work with, and they're only about 30 minutes from my home here in Massachusetts. Anastasia Khvorova, whom I've actually known for quite some time, and our paths have crossed, much like Ken and my paths have crossed in the whole RNA world. She is an oligonucleotide chemist, but has very broad knowledge around therapeutics. In fact, she's been a founder of several companies in the RNA therapeutic space. Vignesh is the person I was talking about, who was the postdoc, and he's now moved on to Comanche Biopharma, which is also developing siRNA. So he's expanding his experience, and he's open to answer questions. Unfortunately, due to certain personal constraints, particularly around his immigration status, he probably can't work with us in the future, but he's open to answer questions anytime. So that's he's still a resource for us to be able to move the program forward. Jonathan Watts is on our advisory board. He's an oligonucleotide chemist and has a broad background, is very, very helpful. Hassan Faki is the person that has been the main driver around a new delivery platform. siRNAs need a little help to get delivered throughout the brain. And so we felt like we needed to license something. He was at UMass, and lo and behold, their work is looking very, very promising in mice. And we've taken now a reasonably broad license to it. Ken, well, we'll talk a little bit later about the license. I think we'll just keep moving on here.
Yeah.
Davut, did you have anything to add about anybody here or Ken?
No, very good.
Good stuff.
Just another point, Monica actually has an honorary degree from UMASS Chan Medical School.
Thanks for pointing that out, Tim.
Yeah, you're welcome. It's an honor, actually. So in our group, you already met Ken and myself. We also have an attorney, Mark Salakian, but I want to point out Michelle Higgin with Pharma Directions, it's basically a boutique. It's basically a pay-as-you-go drug development company. She can have access to all the types of resources that you need: regulatory, toxicology, and even clinical resources. Although that's where Davut comes in on our scientific advisory board. But Michelle Higgin has 25 plus years of experience in drug development.
I might add just on Michelle. So I've worked with her previously. The biotech business can be virtualized to the skill sets that you need, kind of a rent versus buy full time. And her organization provides all of those infrastructure skills. So if you need somebody to help you manufacture, if you need somebody for project management, if you need somebody to help you with regulatory, they have highly skilled consultants within their network. Really an interesting organization, very interesting concept. And having worked with her before at a prior company where basically they helped take it from an idea into phase 2 and now on the edge of phase 3, I know her skill set as a company and as a person.
Great, thanks, Ken. Our scientific advisory board, Davut, who's on the call today, and we're thrilled to have you, Davut, as the clinical expert really on our scientific advisory board. It's really wonderful to have you in Houston, where there are so many connections to the MECP2 duplication community. Harrison Gable, who's an MECP2 biology expert, did a postdoc at Harvard with Greenberg there. John Watts, I've already pointed out his expertise. Vignesh, he was working with us. Again, he won't be specifically on our advisory board, but he'll be able to advise us sort of in an unofficial capacity behind the scenes. And Hassan, who's a delivery expert. I already talked about him. We're going to be working and using a novel siRNA. It's stabilized so that it is not degraded rapidly. But we tried to minimize the number of modifications to it so that it doesn't introduce any untoward toxicities. And this dendrimer technology that we can add readily to it on a DNA synthesizer to provide broad-brain biodistribution. For those of you that are interested in the brief science, siRNA comes in double-stranded. It is double-stranded RNA with certain construction. There's a sense strand and antisense strand. It then loads up something that's called the RISC complex, and the RISC complex then recognizes the messenger RNA, and then it cleaves that RNA into little pieces, and then it can't make any protein. So in much the same way that the ion 440 is targeting messenger RNA, we are targeting messenger RNA, but we're targeting messenger RNA in the cytoplasm, not in the nucleus, which is what ion 44 gapmers would do. So that's one specific mechanism difference. Davut, do you have any particular comments about using siRNAs or for this particular approach?
Rather than ASO or the siRNA, I wanted to say this. The gene therapy is a new field, and we are learning it. Like, for example, if you ask me 2 years ago, the gene therapy is going to be one-time shot, for example, gene replacement therapy. But now I am saying it may not be the case. We may need to redose gene therapy in the every 5 to 10 years, because the first patients who got it, now they are getting like weakening, having some muscle diseases. I'm talking about SMA, spinal muscular atrophy. So they may need to be redosed. So long story short, we are learning on this path as well. And there are a lot to achieve successful clinical trials or the successful results. But so far, my experience, if the families ask, RNA-based treatments are to go at this stage because their safety profile is much better. And also, you don't have a one-time shot. You redose. That's why if you overdose or underdose, you can fix it next time. So overall, that's why I am putting more effort. I recommend like RNA-based treatments. I would say that more safer to go at this stage of the gene therapy world.
And I don't know, Davut, your experiences overall with gene therapy, but of course, the issue of the immunological response preventing the ability to do multi-dosing with gene therapy is an issue right now. Now, it may be solved when somebody comes up with a non-immunogenic AAV delivery technology, but that's not there yet, to my knowledge.
That's mainly, again, I can mention this is the viral delivery is the biggest problem for DNA-based treatments. There's a lot of side effects, okay? And you probably all know that the Rett syndrome gene therapy, high-dose patient died in Europe. So this can happen definitely with the viral delivery methods.
Yes, and this will be treated by the agency almost like a small molecule.
Yeah,
It's not a
different.
small molecule, but
Really,
very,
really important, different division too, as well, probably.
that's correct. It's CEDAR, yeah, not CEPR. So continuing on the theme that Davut introduced there, as far as what are some of the benefits of our particular approach, we believe that siRNAs, not we believe, they've been demonstrated to have significant duration of effect, and they've been able to show this in humans as well. Not in the CNS, there are no, that I've been able to find recently at least, recent searches, no siRNAs that are approved for the CNS and the brain, but multiple of them have been approved for Alnylam in the liver. Based on animal data, we believe that our construct will have 2 to 3 times greater duration of effect than ASOs. With the dendrimer attached, we'll be able to have broad brain biodistribution. And that part of what we're going to be looking forward to raise money to do is that, and we'll show you our plan around that, is to show that in larger animals, such as showing it, so far it's been mice, we'll show it in rats and dogs down the road, which would also provide the tox package to move it into the clinic. We believe that these provide an improved safety and a therapeutic index because usually with siRNAs, they're more potent than your classical ASOs, and you can get the same effect by providing less of the entity itself to the patient.
Tim, I can briefly jump in here. Families may not understand what is biodistribution. So to explain the brain, okay, we have, every human being have around 20,000 genes, okay? And 80% or more of them are expressed. That means they work in the brain. That shows that the brain needs a lot of genes or majority of the genes to work properly. And the brain is not one, like it's a one organ, that's correct, but it controls everything, right? So that's why each segment has different roles. For example, back of the brain has balance, coordination, or kind of speech organization, visual organization, and everything. There is a sensory part of the brain, the motor component of the brain, Some areas just, for example, damage to those areas cause seizure. So when you deliver this gene to the brain, for example, you can do lumbar puncture, or you can go, for example, in the Rett gene therapy, at least the one, the neurogene, you do intrathecal. You go into the brain from here and put into the empty space, fluid-filled space in the brain, and you expect that it's gonna go everywhere. But the reality is not like that. So it doesn't go everywhere as you want. So biodistribution means in the biological environment of the brain, it needs to go everywhere in as needed. That's the point. For example, movement part may need more MECP2 than the control part. That's the reason maybe you don't see too much balance problems. You have, but not that much. So that means that you need to have ideal amount in each area. So that's the hope there with the biodistribution, proper biodistribution, you are going to get everything where they need ideally, or enough at least to some degree.
That's correct, Davut. Thank you for explaining that more broadly. We anticipate that this drug would be administered IT intrathecally, which is in the spinal column, okay? Much like Spinraza is, I mean, exactly like Spinraza is administered right now to SMA patients. So we have some thoughts I'll talk about later about how we can do some research to develop something that can be administered IV, which would be a game changer. But right now, those are just concepts. And for the current product that we've already got a lead candidate for, we would be moving it forward as a product that would be administered intrathecally, again, administered through the spinal column, which would give broad distribution, broad spread throughout the whole brain, okay? So right now, we've incorporated the organization in 24. Thanks to Ken and his work, he negotiated the license with the university. Ken, you want to talk a little bit about that? And frankly, it wasn't the most straightforward thing to do, but you would talk a little bit about the status of it and what we really have as far as a license.
Yeah, well, academic license negotiation is interesting and complex. Monica was very helpful in the process, and we have a full, and I think it's a good terms, very good terms actually, license to the siRNA and the use of the dendrimer exclusively and specifically for delivery of the MECP2 siRNA. More recently, because of the need and question of international filing on the dendrimer technology, we have been able to broaden the license to allow us to add in additional oligonucleotides that could be delivered using the dendrimer to the brain for brain diseases that occur in or are controlled by the brain. We're finalizing that amendment. That's important when we get to the business discussions about the fact that MECP2 duplication is a very rare disease. For those of you who follow the financial markets, rare diseases were an area of extreme focus. But when a disease state is too small, it's hard to get larger financial people to fund those. Being able to broaden that license and therefore paint a picture of a broader, bigger company someday in the future with the technology is going to be very helpful for discussions with, when we get later in the development of this company. Not this week, if you will, but as we evolve, and we have to be aware of the financial markets. I think we'll get to that at the end, but that's the setup on the license. And we've been just had an extremely good relationship with the UMass people. They have been very good partners across the board as we've gone through both the development process and the work that was done by the RSRT previously, and I think will be done in the future. So we'll leave it at that for now, because we'll get to some of the future thoughts as we get to the end of this presentation.
Thanks, Ken. We've already talked about the people that we have in place to move forward the program from a development point of view. We've been able to recruit a good, focused scientific advisory board right now. And we have a research plan that we'll execute with the proper funding that we're trying to obtain.
Yeah. The one thing I might add here is that Tim and I are of a vintage where we have built companies and been in companies before. I've started two from scratch. Literally for the cord blood stem cell company, I was the president, CEO, and sole full-time employee. So we understand what it takes to put the pieces together to build an enterprise. I kind of liken it to Athena springing fully formed from the head of Zeus. We're small, but we have all the components of building a company. So that's how we think about M2DS. What does it take not only for today, but to build it going forward? And so that's why the SAB is put together. We have Michelle, we have a extremely well-known corporate counsel, just putting those pieces together to make sure that in the creation of the company, we have an enterprise that can grow and be funded.
And we have folks like Davut who are, generously, contributing their time right now to guide us on the clinic, towards the clinic, because that's what matters in the end.
Yeah, you have to think about regulatory strategy and clinical development strategy at the beginning
Yes,
of these enterprises.
that's right. And so where are we in the drug development to this? We're really at the preclinical stage. Don't want to try and represent it that we're any further than that. We have very good data. Again, thanks to Vignesh and what he's done and the folks in Anastasia Khvorova's lab. In mice, we've been able to show that we can knock down MECP2, been able to show that we can improve symptoms in a transgenic mouse. So we're really to the point where we need to move the program through what we call IND enabling types of activities, including toxicology. We've identified the place to be able to manufacture the drug. They've made these types of molecules before. The beauty of the dendrimer technology is it doesn't require a lot of special, it can just be done right on a DNA synthesizer where you make RNAs typically. So, and they can make it at quantity, gram scales. It's a company that's out of the Boston area called ChemGenes. But in general, this is a general drug development timeline pathway, probably for MECP2 duplication, much like what's going on with the Ionis trial, probably be able to combine phase 1, phase 2, and get into a significant number of patients and convince the FDA. We would hope for fast track, but that's hope right now. We don't know. We haven't had a meeting with the FDA. I don't want to give you the feeling that we've already had that conversation because we haven't yet.
Yeah, and when you say fast track, it's meaning that we would not necessarily need to do the phase 3 larger trial.
That's what I mean. Yeah, exactly.
Yeah.
So the market size, we've looked at it from 3 different angles. One was based on the Australian data that came up with roughly 1,100 to 1,200 patients. A different angle we took was through X-linked intellectual disability, came up with 1,600 to 2,000 patients worldwide. And then through advocacy organization statistics, probably around 4,000. The only reason I put these up is to point out, again, the comparatives. If you look at what Alnylam has gotten approved for their rare diseases, the first 2 were 150,000 patients. Then the following are probably in the ballpark of what we have for MECP2 duplication, which are roughly 5,000 patients in US and Europe, and same with this nedosiran, if I pronounce it right, about 5,000 patients. So we're probably in that type of ballpark. The difference is that Alnylam already had developed their product with the capability of getting things to the liver on the larger indications, the 150,000 patient population indications. So this will require a completely new development technology for CNS. However, having said that, we have every reason to believe that this would be effective with intrathecal delivery. We'll have to show how safe it is in the rat and dog talks.
I wanted to interject
Yeah.
here again, Tim, sorry.
Mhm.
So some of the families who are talking to me, they are aware that on top of MECP2, I started working on another gene called RAA1 gene. Again, it's very similar to MECP2, too little cause Smith-Magenis syndrome and too much cause Potocki-Lapsky syndrome, which is my former mentor. The frequency of SMS and PTLs, each one is around one in 20,000 or 1 in 15 to 25,000 individuals. And it is very hard to attract companies with that frequency. I don't know why, but at least the Rett world has that aura that drug companies have some interest. But we are very thankful for the MECP2 duplication world, I would say, for the patient society. At least you have like 2 ongoing clinical trials and then other potential approaches like siRNA here. I think this is amazing, just to let you know, for a rare disease such as MECP2 duplication syndrome, having options, couple of options is amazing, just to let you know. This is very, because I am literally trying to get some companies to develop some different approaches or the ASO or whatever for RAA1. No, they are not interested. And the frequency is probably 5 to 10 times more than MECP2 duplication syndrome. So we are lucky in the MECP2 duplication world. I can say this for sure.
If I can jump in for a sec, I think that's in large part due to a couple of things. One is the association, like MECP2 is a well-known gene. Like, there's been lots of money that's gone into Rett syndrome research. There's high-profile researchers working on it who are presenting at high-profile conferences, publishing in high-profile journals. And that link, I think, to, because it's the same gene, and because Rett syndrome is a fairly high profile disorder, I think that's really helped the duplication syndrome.
Yeah, the duplication definitely benefits directly from Rett. As you said, the high profile, like Huda, there was few other crazy scientists at MIT or Harvard. So they are very well-known scientists and they work on MECP2. That's the blessing, to be honest. Yeah.
Yeah. And then the other reason is the hard work that many of the families that are on the call today, fundraised, and we funded, for example, the reversal experiment. Like, that's important. Companies,
Very important
experiment. It was important in Rett syndrome. Adrian Bird did and published it in 2007. It was important for duplication. We knew it was important, and that's why we raised the money and asked Huda to do it. So I think there are these building blocks in place that have been really, really helpful. And a shout out to the families that have fundraised, and to Cure MDS and Pete Anderson and all the families that have worked with him to raise the money. Okay.
This is, sorry, again, jumping in, Monica. So as you said, definitely the initial fundraising by the families is key, one of the very key, because it needs to reach certain level of success so that it's going to attract the companies more. Oh, there's a potential success here. So many of the lab successful trials, they may not be successful in the actual clinical trial. That's why industry is looking at those things. So initial studies needs to be done kind of by the families initially, unfortunately.
Great. Thank you, Davut. Thanks, Monica. So what's our strategy to fund this? We are currently working on filing a small business innovation research grant. We're working with a company that has a pretty good track record in getting these things funded, Alien Tech Transfer. They get about a 50% to 60% success rate on these grant submissions. We're looking to do what they call a phase 1. This is not FDA phase 1 and phase 2. This is phase 1 with regard to the grant process, which is a phase 1 and then followed quickly by a phase 2 for a total of about 2.4 million. We feel like this would allow us to really determine what they call a maximum tolerated dose, as AIM-1 states there, in rats, and then follow up for GLP toxicology in rats and dogs. They have accepted our proposal. Alien only accepts about 20% of the proposals that come to them. So we kind of passed a first bar there. We'll see what happens. We have to get this into them by December 18th for January 5 filing deadline with the government, and then I think we know within about 6 or 7 months after that. So that's one avenue we're doing to move forward with this. Davut has talked about, and Davut, maybe you want to talk a little bit more about this. We are starting to formulate in our minds how we might be able to obtain grants to do initial clinical trials at Baylor. Do you want to talk a little bit more about your vision? And we realize it's just a vision right now, Davut,
Yeah.
about that.
So I looked at the opportunities. As you said, we are still like early stages to bring into the clinical trial. And M2DS is an industry, so that, but NIH has these programs, it looks like, that academy and the industry can collaborate on the early stage kind of studies. And basically, siRNA MECP2 duplication seems to be a good fit for that. So that I will work on it, but I don't know if you know also the current administration, the current NIH environment, the budget cuts for the research and everything. I don't know how it will be impacted, but all I can say is NIH kind of focus shifted from basic science research to more translational or clinical research. So from that perspective, I am hopeful that even if they cut the basic science research, basic neuroscience research, maybe our kind of proposal, this can go, has a chance of getting approval. So we'll see. But this is in the kind of still early stages, but there are opportunities for us to apply. Hopefully that can bring us to some level. Yeah.
Right. Thank you, Davut. We would like to work with the duplication families through RSRT to fund 2 key experiments. However, comparison study of our 1764, which is our siRNA dendrimer conjugate, with ion 440 in the transgenic mouse model, which was done by Vignesh earlier on with a different construct. We need to fine tune that a little bit and put that head to head with ion 440, which is in the clinic in this transgenic mouse model. So that's one study. We would also like to develop at lab scale. This is completely new concept here, and it would be a game changer, frankly, not just for MECP2, but many other diseases where one is administering either siRNAs or even oligonucleotides. We have some concept about how we might develop a construct for IV route of administration. So you don't have to get a lumbar puncture in the spinal column. You could just administer it IV in a vein, which we think that would be a complete revolution, frankly, in how these types of molecules are administered to patients where they need to get into the CNS. And so we're looking for, really looking to put in place a grant to do that through RSRT and through the duplication families. Once we've hit these milestones, we'll need to persuade mission-oriented investors to actually invest in the company so we can move forward the clinical work. Just depending upon work from Davut, as Davut pointed out, funding environment is very interesting. And even if we do get grants to be able to do clinical trials, they have their own speed with which they can really proceed. So we will need to persuade mission-oriented investors to invest in the company for more clinical work and, of course, to obtain FDA approval. Davut or Ken, you or Monica, you want to add anything there at this point?
I think the only thing to add to this is I think we are all aware of how complex the external environment is for funding in biotech companies. I have a line that I've always used, which is nobody has ever invented a cure for the business cycle. So this will change, but we don't know when. And so we have to prepare for the complex environment we currently live in. So for example, when we started this program, pediatric vouchers were available. And that was actually a funding mechanism for companies such as ours, because you would hope to get a pediatric voucher, which speeds up the approval process in the FDA, but you wouldn't keep it yourself. You'd sell it to somebody for literally $100 million plus or minus, and you use that funding. That doesn't exist right now. And so it may or may not exist by the time we are further advanced as a company, but it just is one more level of complexity that we have to think about and address as we evolve this company.
Davut or Monica, anything?
No.
I wanted to say this IV route, now running the ASO trial, the SMA, the initial approval of ASOs, those kids are intellectually, mentally intact, right? So you can tell them stay still, and they don't move because of the muscle disease. They don't move too much. So you don't need to sedate them for lumbar puncture. So that is the beauty of that disease. But for MECP2 duplication, you need to sedate them. You need to sedate because they won't listen to you, or they won't, stay still when the needle goes into your spinal column, right? So it is a challenge. These kids have trouble with waking up from anesthesia. So yeah, definitely an IV route will be a great achievement, milestone for this condition for sure.
Thank you, Davut. So we've already talked a little bit about some of these factors that will require what we call mission-oriented investors, a very small market for the rare disease, and all the approved products are in the liver and none are in the CNS. We've already talked about the fact that we've expanded our license so we can go beyond just MECP2 if we want to develop the company more broadly with MECP2 as the first indication. I did a little looking. The FDA voucher system that Ken was referring to, as he pointed out, is not currently renewed by Congress. However, apparently there's something called Give Kids a Chance bill to reauthorize the pediatric disease program, has passed the House, but is now awaiting the Senate.
I think we need to explain what that voucher is, because most people
Okay.
on the call won't know what that is. Can you explain it to him?
Sure. The pediatric voucher really provides for a much faster time to market for a given product. That's the bottom line of what it does.
But it was a perk, right? It was a perk that the FDA would give a company that had managed to get a drug approved for a rare disease or pediatric disease.
Yeah, it actually started off with tropical diseases. That was the idea. It got picked up within a year to be used for rare diseases, primarily coming out of the cancer community, and a person named Nancy Goodman who pushed it aggressively, and then it became more broadly utilized in rare diseases. It doesn't exist right now because it was not reauthorized, and they're obviously pushing hard for it. There is a voucher program right now that exists, which was just announced in the last couple of weeks for 9 companies, a couple of which had applied and a couple of which were chosen by the agency, but it only speeds up the specific drug that the company has. So it's not a financial tool, if you will. It is a regulatory tool. And we'll see how this evolves over the course of time. Not clear yet.
Basically, you get moved to the front of the line. I mean, to put it in simplest terms possible, right? At the FDA.
Yeah, and companies, if they got one, if they got a drug approved, they got this voucher, they could keep it for themselves to use it on another drug, or they could sell it. And for a while, these vouchers were selling between $100 and $300 million.
Supply and demand.
So
part of the deal that we structured with UMass at the time we structured it was that they were going to get a percentage of whatever we might get from a pediatric voucher if we ever got it. So that was part of the basic structure of the initial deal. Whether or not that becomes reality or not as both a funding mechanism and an incentive mechanism for whatever we want to do as a company is just completely unclear at this time, to be blunt.
Right.
But we're hopeful. I mean, I think logic says that there is political pressure, but I think, as everybody's aware, it's on a long list of political pressures, is a nice way of saying it.
Great. So what's the competitive landscape for MECP2 duplication? The two biggest competitors to what we're doing, or we're competitors to them, however you want to look at it, you all know about Ion 440. Most of you probably know about Huidagene's CRISPR-based RNA editing, the H204. And they will go through what we view as some of the pros and cons of our technology with respect to theirs. Ionis, antisense, they're in the clinic as of late last year. It's using technology that has human experience. It's titratable, as Davut pointed out earlier. It's a large company that's based on antisense, so they have the resources, they have all the people in place to do it. The problem that we see, it's not a problem. Antisense does require probably more frequent dosing than siRNAs do, just typically, classically. They tend to be less potent than siRNAs. And for theirs, they have to depend upon the actual oligonucleotide backbone to really provide the brain distribution. They don't have a particular moiety that they attach to or change in structure such as Atalanta to be able to produce a broad brain distribution. Huidagene is in the clinic as well at the end of last year. It's a one-time dosage. It has the disadvantages that Davut was talking about earlier. It has to be delivered by an AAV, so there's immunogenicity associated with it. And you, from what we understand, you currently cannot redose it. So you can't readily titrate it for the patient, which is the great point that you made, Davut. Do you want to talk about that anymore, or did you think you made all the points you wanted to make about that, Davut?
And maybe questions maybe there, yeah, but yeah, for now that's fine, I guess.
Okay. So our technology, we think it's more potent, as I pointed out. We believe it will have, and in mice, it does have a longer duration of effect. It's titratable. We have a delivery modality that we attach to it that provides, again, broad distribution through the brain. And it follows after products that are on the market using siRNA technology. So it's not a completely brand new technology. So we feel like it brings novelty at the same time as utility as compared to some of the other technologies. Our cons are we're behind the other programs, right? We're in the preclinical only, and our delivery modality has no human experience. It is animals only. A priori, from the structure, one would not predict toxicology, but you have to show it and prove it. Any comments here, Davut or Ken or Monica?
Yeah, I just, from a parent family perspective, right? We don't think of it in terms of competition, right? The more options, the more companies, the more modalities, the more delivery technologies, the better. And that's what we're trying to do for Rett. And I think that's what we need to do for duplication. We need options. They may or may not all move forward. Some may work better than others. So options is the name of the game. Of course, from investors, and if they're doing diligence on a particular disease space, it's very important for them and critical to understand what the competitive landscape looks like.
And let's be realistic. For those of you who follow drug development, being in the clinic does not guarantee that your drug will succeed and be approved.
Right.
Tim and I have, for lack of a better term, battle scars from that life.
Yeah. Plus, as Monica points out repeatedly, and has experienced, frankly, in the Rett space, the corporate priorities change often with organizations, and they may abandon a molecule. I'm not saying that Ionis is going to abandon it, but priorities change constantly within the organizations. So,
yeah, I mean, I think of it. There's science risk, right? From basic science, and does something work? Does it have toxicity? Does it, are patients getting better? So there's the science risk, but then there's also the business risk, right? And you're competing, every program basically is competing against other programs that's on the company pipeline or new programs that a company might be bringing in. So there's always this competition at every company, for whatever it is they're progressing. Another program may be progressing quicker or better or whatever. So there's business decisions. We saw that in Rett when our first gene therapy that started off in a company called Avexis, and then Avexis was acquired by Novartis, and Novartis dropped the program. And when that happened, it was pretty traumatic for our community. Fortunately, we didn't have all of our eggs in the one basket. So there were other gene therapy programs, and there were lots of other modalities as well. But this kind of stuff happens all the time. So that's why you need options.
And like one thing maybe there, Monica, it's weird because we showed several times that MECP2 duplication is not a single or everyone has the same mutation, right? Same for Rett syndrome. To give an example, for example, some of the Rett patients, they respond to, or some of the gene therapy patients, for example, they respond to Spinraza better than the other mutations maybe, or there is other genetic modifiers, whatever. I can say this, for example, some of the patients for Rett syndrome, they respond well to Davut. They knew it's not a genetic treatment, I know, but everyone is similar. Like you may respond to Tylenol better than me. So that's why different options may target different and may have better kind of improvement. Oh, this type of mutations may need this approach, or this type of mutations may need this approach. So options are always very good, important. Yeah.
Great. Thank you, Davut. Thanks, Monica. So here are some of our, as Monica aptly pointed out already, the backup strategy in case other technologies don't advance. Our first step, as I already pointed out, is to do single ascending dose in wild type rats followed by multiple ascending dose. Could be considered rodent tox in that first, but we wanted to understand what the amount is that really reduces MECP2 and causes any particular CNS effects. So we want to really move our current funding as we want to move the program quickly so we can do formal IND enabling activities, okay? And we also want to develop something that can traverse the blood-brain barrier just by intravenous administration instead of intrathecal administration, as Davut was pointing out. These are broad brushstrokes, okay? This early stage takes probably 500,000. We're actually asking for the grant for the two experiments I talked about, around 300,000. We'll be writing a letter of intent to RSRT for the transgenic mouse comparison, as well as for doing some early work on this intravenous route of administration. It usually takes $1 million to $2 million in order to get going with our SAD, but to really get the rats and dogs, again, we hope that we get the grant but again, that will not cover all of the talks. It probably will need about an additional $1 million on top of the 2.5 that we're going to get from the SBIR. And then to build the company, we'll need a minimum of 10 million just to probably get going and really move things into the clinic, produce enough clinical trial material, et cetera, and get all the regulatory pieces in place, et cetera. Of course, you all know about the disease way better than I do. And we appreciate very much your sacrifice for your family members with this disease. We also understand that your money is the most expensive and sacred money that there is, frankly, when it comes to doing these types of things. And so we just express gratitude and appreciation for what you do for the community. I pointed out we're looking for about $300,000 to conduct these critical experiments we're talking about. We're also looking for investors in the company. If you know people, your network is broader than those that are just on this particular call. There are people that have buckets of money for making investments. There are people that have buckets of money for making philanthropic donations to like RSRT. We realize that they are not necessarily all the same people, or the same people don't always have the same buckets of money for those two activities. So, but we again appreciate your attention today, and here's some contact information. Ken, maybe the best one is not there for you. It's
Yeah, it's KMOC at M2DSTX.com.
yeah.
So just KMOC at company name TX for therapeutics.com.
Ken has lots of emails.
And you can just email me. I'll direct the emails.
Yeah.
To most
Monica
people.
knows where everybody is.
Yeah.
She knows where we live. Yeah.
Just want to thank you for your attention and appreciate the opportunity to speak with you all today. The contact I've had with the MECP2 duplication community in the past has always been delightful. So thank you so much.
And we are
Happy to
delighted
answer
any
questions.
Yeah.
I don't see any questions, but if you have any, now would be the time to ask them. And if you think of questions later, you can email me and we'll make sure to get back to you with replies. Well, Ken, I think you must have done a very good job because there are no questions.
Tim. Yeah, Tim. Good job.
Tim, I'm sorry. Ken and Tim and Davut. All
And right. All and you, Monica.
Thank you all.
Hold on. There's one
A
question.
question just popped up.
Why is it that children have trouble waking from anesthesia? Davut?
Sure. I think the main reason is the low tone that these kids have, or different stages. Let me say that as the families know, in the early stages, the first few years, 6 years, 7 years, they have low muscle tone. So when they have low muscle tone, their breathing muscles, their throat muscles are not working very efficiently. And that's why when they are sedated, they're not perking up enough to breathe on their own and everything. And as they get older, turning puberty, the seizures on board, they are on multiple medications. And we all know that they are very drugged from those medications, plus the burden of the seizure. I think that complicates the situation. So that's why different age groups, that's the reason they don't tolerate. I would say that relatively younger age, they tolerate the sedation better, but as they get older, definitely it is much harder.
Okay. We have a question. Are you going to innovation centers at children's hospitals for funding?
Have not to date. It's an interesting question, but unless they have a reason to be particularly interested in this disease state, it's going to be hard because we're obviously competing with all the other activities that are ongoing right now.
And another question, what's the minimum amount to invest?
Well, Monica, we talked about whether or not it's going to be people donating to RSRT so they have the tax benefits and then some sort of grant structure with the company. I think that's still, you may have some specific thoughts. I haven't thought about a minimum structure, but
well, we need to think about that, an investment in the company, what's the minimum threshold versus a philanthropic donation to RSRT to fund a particular project. So we'll get back to you.
Yeah, I think that I think that the only way we could do at this amount of money, a couple of $100,000, if you will, it would probably be structured as what's called a safe note, because I think to go through the legal paperwork of creating an equity investment becomes actually too costly. That's why they created. So safe note is, I forget the exact what safe stands for. It does stand for something, but it's a widely used tool where the actual price of the financing and the structure is set at a later date when a larger amount of money is received. So it reduces the upfront costs, which can take away from the ability to do the research that you want to get done.
Yeah.
Tim, I don't know if you want to elaborate on that from your
No, that's great. Thank you.
So it would probably be done. The answer is, if we have a threshold of interest, we'd probably structure it as a safe note, would be the way I think about it.
Okay. Well, it's 1 o'clock. That was beautifully timed. Thank you everyone for joining. Enjoy the weekend. We'll